Hello biology students, and welcome to video 8, the final video of the organic chemistry unit. So we finally reached the end, and we're going to talk about our last macromolecule, which is, of course, nucleic acids. So nucleic acids, just like every other macromolecule we've discussed, have carbon, hydrogen, and oxygen. Like proteins, they also have nitrogen, but unlike any of the others, they have phosphorus. So that's kind of the defining atom or element type would be phosphorus. So if something contains phosphorus and it's an organic molecule and it's a macromolecule, you know it's a nucleic acid. These are very important to some functions. They serve as instruction for making all proteins, okay? So for every protein, remember we have thousands and thousands of proteins in our body, the instructions to make those proteins come from nucleic acids. And they also store and transmit, so pass on, all of our hereditary or genetic information. So these are pretty important molecules we're talking about here. Okay, so the polymer of a nucleic acid is, I know, very hard to remember, is called a nucleic acid. So that's nice and easy. Nucleic acids, the polymers are nucleic acids. There are two examples, I should say, of polymers. So DNA, which you've all heard of, I know, which is found in the nucleus of the cell. And RNA is the other main polymer, and that's found in the cytoplasm of the cell. And the monomer, so those are the polymers, the monomers are called nucleotides, okay? So just like with every other macromolecule, we're going to use a dehydration synthesis reaction to link together nucleotides to form a nucleic acid. So let's look at what a nucleotide looks like. So here's a nucleotide. Remember, this is the monomer of a nucleic acid. And the nucleotide has three main pieces. It has a 5-carbon sugar. You can see a 5-carbon sugar right here. Remember, I have a carbon at each of these little bends. So 1, 2, 3, 4, 5. That's my 5-carbon sugar. It has a phosphate group attached to the carbon right there. And then off this carbon over here, it has what we call a nitrogenous. And nitrogenous just means nitrogen-containing base. Okay, and you do have to know the structure. So I have a 5-carbon sugar with a phosphate group on one side and my nitrogen-containing or nitrogenous base on the other. So let's look at the two different polymers. So the first polymer was DNA, okay? So in a DNA nucleotide, so in a monomer that makes up DNA, the 5-carbon sugar that we had is a sugar called deoxyribose. Right? And actually, DNA, the name, actually stands for deoxyribonucleic acid. So they're saying it's the nucleic acid that has deoxyribose as the sugar. It has a phosphate group, like every other nucleotide would have. And then the nitrogenous base, okay, that nitrogen-containing base, in DNA could be one of 4 things. It could specifically be adenine, which we symbolize with A. It could be thymine, which we symbolize T. Cytosine, which we symbolize C, and guanine, which we symbolize with a G. Okay, so over here, I kind of have one part of a DNA molecule. You can see I have my 5-carbon sugar, which is deoxyribose, attached to my phosphate, attached to my nitrogenous base. So this would be one nucleotide. And then through dehydration synthesis, I can link it to a second nucleotide, and to a third, to a fourth, etc. So what DNA actually looks like is DNA is actually a double helix. So it's 2 strands that have been twisted together. So you can see I have one strand here. Can you see how I have a nucleotide linked to a nucleotide, linked to a nucleotide, linked to a nucleotide? And that strand is bonded to a second strand on the other side. Okay, so there's 2 strands of these nucleotides twisted together. That's DNA. RNA, okay, so RNA is also a polymer, so it's also made up of the monomers nucleotides, but the monomers in RNA look a little bit different. So in RNA, that 5-carbon sugar is not deoxyribose, it's just ribose, which again makes sense because RNA stands for ribonucleic acid, so it's the nucleic acid made with ribose. The monomers, they have a phosphate group, and they also have a nitrogenous or nitrogen-containing base. But in this case, the nitrogenous base is either adenine, which is symbolized with A, cytosine, which is symbolized with C, and guanine, which is symbolized with G. So those 3 are the same from DNA and RNA. But instead of thymine, instead of T, RNA has this base U, uracil. And where DNA was double-stranded, RNA is going to be single-stranded, okay? It's not going to be two strands. Look at a picture, you can see I have my nucleotides, but it's only one strand. There's not a second strand there. So if we look at there are four main differences that we can see between DNA and RNA. They have different sugars. RNA has ribose, DNA has deoxyribose. And I want you to look at this picture for this one over here. In case you were wondering where these names came from, because I always like to give you, word root hints. I think it helps you remember. Deoxy, that little prefix there, means without oxygen. So if you compare deoxyribose with ribose, the only difference is deoxyribose is missing an oxygen right here, and ribose has an oxygen. That's the only difference. So they're very similar. They have different sets, so that's the first difference. The second difference is they have different sets of nitrogenous bases. So the A, G, and C are the same, but RNA uses uracil, where DNA uses thymine. They have different strandedness, so DNA is double-stranded, RNA is only single-stranded. And then they have different locations, and this got cut off. So DNA is located in the nucleus, and RNA, I'm going to have to add this in, it got cut off. RNA is located in the cytoplasm. Good. Okay, so we said earlier that one of the functions of a nucleic acid is to determine the structure of a protein. So basically the way this works is a gene is a section of DNA that tells you the order of amino acids that should go in a protein. So basically the order of nucleotides in DNA dictates the order of amino acids in a protein. And the way it works is that because DNA is so important, it's located in the nucleus, okay? And it turns out we make proteins in the cytoplasm. So what RNA is this intermediate molecule that kind of reads the DNA, and then it brings the DNA message to the protein, to the cytoplasm, so the protein can be built. Okay, so RNA is an intermediate between DNA and protein, so that way the DNA can be kept safely in the nucleus and the protein can be made somewhere else in the cell, in the cytoplasm. So that's it for the organic chemistry unit, and I'll see you in the next unit, which is going to be all about the cell.